several of you have heard me lecture on HIV in the past from a number of different perspectives. And one of the amazing things is that one could, for example, have a lecture on immunology. But what I'm going to try and focus on today is the issue of the evolution of the virus, which is something that happens both in terms of broadly changes in the virus that can be detected in a population of infected individuals, but is also a process that occurs within the body of a single infected individual as well, and in fact is very closely tied to the way in which HIV is able to cause disease in the body of a single infected human. So what is quite clear is that at the level of both the human species, if you will, as well as at the level of the individual, HIV is clearly an evolving threat, and in fact the process of rapid molecular viral evolution is in some ways one of the crucial strengths of the virus and what allows it to really be such a powerful enemy of the human species. So before I get into that, for those of you that have not heard me lecture before, what I'd like to do is to give you sort of a quick update on where we stand today when it comes to the HIV sort of virus as far as the epidemic is concerned, both in the United States and globally. So that as many of you know, HIV remains a very significant concern in the United States, but as recently as 1996 to 1997, there was a remarkable decrease in the number of deaths associated with AIDS in the United States, a decrease of some 42%. And if some of you remember, you may in fact have seen the cover of Newsweek that posed the question of whether or not this was the end of AIDS. And what we now know is that this remarkable decrease in the number of deaths associated with this disease was really due to the emergence of what is known as combination therapy, which today remains the gold standard for how patients with HIV infection are treated, particularly in the United States and in the West. So that what we're talking about is a combination of usually 3 different anti-HIV drugs which have a remarkable impact on decreasing the amount of virus present particularly in the bloodstream of the infected individual. And that what this therapy did in 1996 to 1997 is dramatically impact the number of deaths associated with the disease. And in fact, at that point, it was thought that the virus potentially had been licked, that possibly we had a combination of therapies that would allow an individual to live to survive with HIV. Sadly, that is now known to not be the case, and that in fact, after 2 to 3 years, resistance emerges to combination therapy, and that this resistance is closely linked with the evolution of the virus in the body of the infected individual. So that as of last year in the US, there were more than 1 million men, women, and children infected with HIV in this country. Last year, there were more than 45,000 new infections. And interestingly enough, of new diagnoses, women accounted for more than 30% of new diagnoses in US adults. So as many of you know, HIV infection and AIDS was initially identified among gay men. And for quite a significant period of time, for almost a decade, the vast majority of new infections were in men. What we now see in the United States is that almost 1/3 now occur in women. And so what we're now starting to see is, in fact, in the US, an increasing incidence of HIV infection among women. What is also quite striking, however, is that if we look at the rate of infections or the rate of new AIDS cases based on race or ethnicity, what we see is strikingly different. That in fact, among new AIDS cases in men, 60% last year were in African Americans and Hispanics. Among new AIDS cases in women, 80% were among African Americans and Hispanics. So that what this means is that AIDS is now the #1 killer among African American women in the age range of 25 to 34. A striking statistic. Furthermore, HIV incidence among African Americans is now 8 times higher than among Caucasians. So that what is quite striking is that while the total number of new AIDS cases per year in the US have held relatively stable for the last 5 to 8 years, if you break it down according to racial groups, the number of new cases among African Americans and Hispanics are increasing dramatically, and that this is a point of very serious concern. So that while the epidemic in the US may appear to be relatively steady in total numbers in terms of the number of new infections per year, which in and of itself is not a good thing, if you break it down in terms of racial group, it is galloping out of control, particularly among African Americans. And we could spend quite a bit of time talking about why that might be, but that is not the focus of our discussion this evening. In terms of the epidemic, globally, the numbers are far more serious and far more dire, as I'm sure most of you are aware. At this point, more than 40 million individuals are conservatively thought to be infected worldwide with HIV. Roughly 5 million alone were infected in 2005 at a rate greater than 11,000 per day. Of those 11,000, 1,500 were in children younger than 15 years of age, and roughly 10,000 were in individuals in the age range of 15 to 49 years old, half of whom are women. So what this means is that globally, half of new infections are occurring in women around the world, so that there is gender equity when it comes to HIV infections. At this point, it is thought that roughly 1% of all sexually active adults in the world are now infected. That's a relatively conservative estimate. And based on how those individuals discover that they are infected, in other words, based on the discovery of their HIV status by accident, by their visiting health clinics for other reasons, it is estimated that globally 9 out of 10 or 90% don't know that they are HIV infected. So that in terms of their ability perhaps to try and limit the risk of passing on HIV infection, the fact that 9 out of 10 don't even know they're infected may have a dramatic impact on whether or not they have any inkling of what they should do to not pass on the virus. Last year, more than 3 million individuals in the world died from AIDS. More than a third of them were adult women, and roughly a fifth were under 15 years of age. So that clearly children are a significant percentage of those which are susceptible and exposed to HIV and those that are dying from this disease globally. So that if we look at the global distribution of infections, what we see quite clearly is that Africa has the lion's share of HIV infections, almost 25 million of the 40 million. What is also quite interesting is that South and Southeast Asia, which includes India, has some 7.6 million infections. What is quite striking is that by current estimates by the World Health Organization and by the CIA, unless something is done about the epidemic in India, India alone by 2015 will have 20 million HIV infections, such that by 2015, India alone will account for half of the current global total. So that what we can see is that even though these numbers are quite dire, in several parts of the world, there is in fact a grave risk of dramatically steep increases in HIV infections if something is not done at the governmental level. That is true of India. It is also true of parts of Eastern Europe, of countries that were once part of the Soviet Union, where, for example, intravenous drug use is on a very steep rise, and HIV infections are rising quite steeply as a result as well. But what is quite clear, though, is that sub-Saharan Africa is being decimated by the epidemic. In sub-Saharan Africa, roughly 8% of 15 to 49-year-old individuals are infected. In Botswana, Zimbabwe, and Nigeria, 25 to 35% of all adults are now HIV infected. In South African provinces, 25 to 45% of pregnant women that seek health care at free clinics are HIV infected. So that if you look at the continent itself, what you will see in terms of the seroprevalence rate among high-risk urban populations, those parts of our sub-Saharan Africa that are in red represent a seroprevalence rate in those populations of 25 to up to 40%. So that, as you can see, there is a dramatic sort of impact of HIV infection in much of sub-Saharan Africa. As one would expect, this has had a dramatic impact on such measures as the average lifespan in many countries in sub-Saharan Africa. So that if you look at the life expectancy in 5 African countries from 1970 to what is projected in 2010, looking at Zimbabwe, Zambia, Swaziland, South Africa, and Botswana, what we can see is that the gains in life expectancy from the 1970s into the mid-80s, into the early 1990s, have been more than reversed by the impact of AIDS and HIV infection, which really hit Africa hard starting with the mid to late 1980s. So that what we can see, for example, in countries like Swaziland, life expectancy has fallen back to the level of 30 years of age, which is essentially what you would have expected in the 1960s in Swaziland. So that at this point, there are more than 20 African nations that the World Health Organization have defined as having suffered severe human development reversals in this past decade of AIDS, primarily in terms of health, in terms of education, in terms of economic wealth, broadly across the society, and in terms of life expectancy as well. So that what we can see from this very alarming graph is that quite clearly there has been a dramatic impact of this disease on the societal level, and that Africa is being decimated by this disease, and that unfortunately there are very few bright spots in terms of the control of this disease in Africa. One exception, for example, might be in Zambia, where we see, for example, a slight upswing that is based on active governmental intervention in terms of AIDS education. Unfortunately, most of these countries, in particular South Africa, has not responded like the government of Zambia, and we expect that things will only get worse in these countries unless something is done very soon. In countries such as Uganda, there has been, in fact, and parts of South Africa, there has been a decrease in the number of new AIDS cases, of new HIV diagnoses, but sadly the reason for that decrease is that the majority of the sexually active segment of the population is already infected, so that the disease has saturated those individuals that are now most susceptible to it based on sexual transmission, which accounts for the vast majority of transmissions in Africa. So that clearly it is a very serious disease, and the World Health Organization for a number of years now has described it as the single largest, the single most serious threat to human public health that our species has ever encountered. And so I think it is not an exaggeration, and it is clearly a major threat that must be dealt with, and that will have marked sort of ramifications in terms of global health and also in terms of the global economy and political stability as well. So what is it that causes this disease? And what we know, of course, is that it is the human immunodeficiency virus, type 1 particularly. And what we're looking at here is an HIV-infected human T cell, and these little spheres that you can see covering the surface of this hapless infected T cell are in fact new HIV variants which are budding on the surface of this T cell. And so what we have is clearly a very powerful adversary, and a powerful adversary that over time destroys the human immune system. What is remarkable about this adversary, however, is that it also represents one of the most striking cases, and actually case studies, in which you can study the process of evolution. So that the evolution of HIV And in fact, the evolution of the disease that we know as AIDS, acquired immunodeficiency syndrome, is a critical hallmark of this disease. And in fact, understanding the evolvability of HIV, how rigorous and how powerful it is in terms of evolution, is a critical part of understanding how our immune system interacts with this virus. But in some ways, it is not just a question of how we interact with the virus. Understanding the evolution of the virus also gives us insight into the origin of the virus. It gives us profound insight into how the virus is able to acquire resistance to a range of different types of anti-HIV therapies. And in fact, I'm sorry to say that every single anti-HIV therapy known is eventually subject to failure based on viral resistance. And so the evolvability of the virus is a crucial part of that. And I don't know if we'll have time to talk about it tonight, but the evolvability of the virus also has a profound impact on the pathogenesis. In other words, how the virus causes disease in the human body. And so what I'm going to try and do is that it is a remarkably complex virus, it is a remarkably complex disease, but I'd like to sort of jump around a little bit across these three areas, hopefully all three areas, in which the evolvability of the virus plays a crucial role in this process. So the first has to do with the origins of HIV. And as one might imagine, the societal context of AIDS produced a whole host of early theories on what its origin might be. I'm not going to go into the more way out theories, like the fact that it's from outer space, the fact that it's a bioweapon gone awry, the fact that it is some sort of conspiracy on the part of the government. I'm not going to go into those theories because I think they're interesting to discuss, but they really don't give us much scientific meat to talk about this evening, particularly in the area of evolution. Where evolution, however, does play a major role are in the context of some major questions. Like, for example, is this disease a new one? Or in fact, is this an old disease that has been present perhaps for hundreds of years in humans and somehow we never recognized it before? The question of where did this disease first arise? In the United States, it was originally identified among small cohorts of gay men on the West Coast and in New York as well. Is that where AIDS was born or not? And since we know that HIV causes this disease, where did the virus come from? What is the source of this virus? And until about 5 to 6 years ago, we could have spent the whole evening debating these three questions with some very interesting insights and possible hypotheses that could answer each of these three questions. As of around 1999 to 2001, however, some very clear pieces of scientific data have emerged that allow us, to the very least, answer the third question, which has the kernels of the answers for the other two questions. And so what we know with relative certitude is that HIV is derived from a simian virus, a virus called the simian immunodeficiency virus, which is actually a family of viruses. And we're looking at images from the electron microscope of a virus that infects African green monkeys. So this is in fact a family of viruses. There are several strains of SIV that are specific to certain species of monkey. So for example, with SIV AGM, it infects the African green monkey. With SIV MND, it infects the mandrill ape. And with SIV SM, it infects the Sooty Mangabey monkey. So that for a particular monkey, we have a particular SIV. Now, these SIVs have been known for several decades, but what was striking about them is that under normal conditions, each strain only infects its specific species and does not infect humans. And so the question was, is it possible that HIV might have been derived from SIV, because in fact the viruses are remarkably similar, with the only difference being sort of one or two key portions of key viral genes. So is it possible that there was some derivation? But up until 2000 to 2001, there wasn't any clear evidence for that, where we could say with any certitude that in fact there was a lineage relationship phylogenetically between the SIVs and human immunodeficiency virus type 1. One could ask the question then, are any of these monkeys susceptible to HIV? And in fact the answer is no. In fact the only other primates that can be infected with HIV, which is in fact very telling, are chimpanzees, our closest relatives. So that only chimpanzees can be infected with HIV, and this was known since the mid-1980s. So the real breakthrough came with the discovery of a particular strain of SIV that is nigh identical to HIV. And it is a strain of SIV that was found, now unsurprisingly, in a particular subspecies of chimpanzee, Pantroglodytes troglodytes. So that in 1999, SIV-CPZ, as it is called, was isolated from Pantroglodytes troglodytes in captivity. It was found to be nearly identical to HIV-1. And you have already heard a number of discussions about the construction of phylogenetic trees. What was found was that based on sequence comparisons between HIV-1 and SIV-CPZ, it is thought that it probably entered humans around 50 years ago. And what is quite interesting is that all of the known strains of HIV-1, it is now thought can be traced back to 3 sort of cross-species transmissions of this virus from Pantroglodytes troglodytes into humans. So that everything that we have seen today, all the different subtypes of HIV, of which there are 11 major subtypes, all of those can be traced back in all likelihood to 3 ancestral strains of SIV-CPZ. Now, this subspecies of chimpanzee is found in Cameroon, West Equatorial Africa, and Central Africa. And unsurprisingly, the natural habitat directly coincides with the geographical pattern of the earliest HIV-1 outbreaks in West Equatorial Africa. So that what we know, what we're quite convinced of, is that HIV-1, the epidemic of HIV-1, is in fact a classical example of what is known as a zoonosis. In other words, the transfer of a pathogen from a non-human species into humans. And that a great deal of effort has gone into trying to understand what is SIV-CPZ like in chimpanzees. And an interesting question is, do chimpanzees become ill from being infected with SIV-CPZ? And interestingly enough, the answer is no. Pantroglodytes troglodytes appears perfectly able to live a healthy life while actively infected with SIV-CPZ, or the equivalent of HIV-1. So a great deal of effort has gone into trying to analyze different isolations of the virus in different wild Pantroglodytes troglodytes. And based on comparisons of different SIV-CPZ isolations, it is thought that the virus has probably been in chimpanzees for thousands of years, based on the diversity of SIV-CPZ that is present in Pantroglodytes troglodytes. Now, I have to tell you that the efforts to analyze these so-called wild isolations of SIV-CPZ are non-trivial. Because I should tell you that Pantroglodytes troglodytes is an endangered species of chimpanzees. So it's very difficult to, if you will, capture them in the wild. There are A, not a lot of them. B, they live high in the canopy of the rainforest in West Equatorial Africa. And so a lot of effort, years of effort, went into, believe it or not, developing methods of isolating the virus from fecal samples on the forest floor. So Beatrice Hahn has led this effort, and she developed methods to first identify pan-troglodytes fecal samples, and then be able to isolate virus in a sufficiently intact form that one could genotype it and analyze the gene sequence with great accuracy. So fortunately, thanks to her tremendous efforts in West Equatorial Africa, we have been able to type a number of isolations of this virus. And so it's pretty clear that pan-troglodytes has had this virus for quite a long time. And in fact, it may be a beautiful example of the evolution, if you will, of a relationship between the pan-troglodytes immune system and SIV-CPZ. So the question is, how did this virus get into humans? Well, as one might imagine, high quality sources of animal protein are a very important part of the human diet in West Equatorial Africa. And for hundreds of years, as far back as anyone can remember, humans in Africa have hunted monkeys and chimpanzees as a source of high quality protein. So it is thought that in the course of hunting, in the course also of butchering for food, pan-troglodytes, probably cross-species transmission have been occurring for several hundred years. It would not be surprising at all, because one could imagine in the process of capturing a pan-troglodytes, you might get scratched or bitten by the animal. You might have a wound that's exposed to blood from the animal in the process of butchering. So it's not hard to believe that transmissions across species were occurring for hundreds of years. But what is very likely is that until relatively recently, until the past 40 or 50 years, infected individuals probably rarely left their local villages or local region. What that meant is that infection was probably confined to small villages, to rural areas, because there wasn't any easy means of infected individuals traveling to then pass on that infection to other individuals in urban centers. What changed that? Of course, economic development has changed that. So that what we see now is the movement of infection from rural areas into urban areas. And thanks to the emergence of plane travel, the movement of infection from Africa to the United States, to Haiti, to parts of Western Europe as well. So this continues to be sort of one way in which infections are transferred from pan-troglodytes into humans, and from humans more broadly into the human population globally. Yes?
The difference between SIV in the monkey versus the HIV in the human. Are you saying they're identical viruses?
They are essentially identical, yes, yes. So that I refer to it as SIV-CPZ when it's in the monkey or isolated from chimpanzees. I refer to it as HIV-1 in terms of its transmitted form in humans, from human to human. But they are virtually identical.
So do they think that originally these chimpanzees did show illness from this virus and have evolved to sort of live in symbiosis with the virus?
Yes, and so in fact that is where the perspective on trying to understand how pan-troglodytes lives with its version of HIV-1 is so important. And in fact, this may give us insight in a rather disturbing way into how it is such a devastating enemy for us to deal with. Because what we're dealing with is a pathogen that has evolved with a primate immune system for thousands of years. And there is something about that primate immune system that allows it to control, to contain viral replication, we believe. The research into the pan-troglodytes immune system is very actively going on right now. Then lo and behold, the virus jumps species, encounters another primate immune system that has never seen it before. And so we don't have the benefit of having evolved whatever mechanism it is that pan-troglodytes has evolved over thousands of years to handle the infection, to contain viral replication, so the virus can go wild. So that an interesting way of thinking about this is that the virus has evolved under what is probably significant systemic pressure in the body of every individual in terms of the immune response to the virus. And the pan-troglodytes immune system does recognize the virus, it does fight the virus. And so one can imagine that when such a pathogen encounters a new immune system that has not evolved, whatever mechanism that might be, the consequences for that immune system are clearly devastating. Well, you request.
Were there any molecular studies done on the T cells in this gene to see if they have the same exact CD20 or whatever receptor or CCR5 that some humans don't have?
Right. So the question was whether or not studies have been done on the T cells of these chimpanzees, of this subspecies, to see if they do have the same receptors for the virus and do they become infected in the same way. So far, the preliminary results say yes. Yes, they do. So their cells do become infected. The virus does replicate. It's not clear how it is that the chimp immune system manages to maintain its capacity and maintain normal cell levels in the face of that level of viral replication. We actually don't know. One possibility is that the regenerative turnover time of the immune system is much faster in the chimpanzee. And oh yes. So it's not as if the virus hangs out in the chimps and does nothing. It does exactly the same stuff that it does to us, but somehow the chimpanzee immune system controls it. So there's some evolved mechanism for control that we don't know what it is yet. David?
Is there any evidence that chimps may have undergone a population crash in the past?
Yeah, so the question was, is there any evidence that the chimpanzees have undergone some sort of population crash? We don't have the evidence yet, but that is being explored. One of the problems we're facing is that we don't have very many of them. The wild population is very difficult to access, and that is the population we need to access. We also don't have, if you will, samples that go back a very long time, hundreds of years ago, in terms of trying to compare hundreds of years ago what things looked like in these chimpanzees. So that really remains a challenge for us. But the expectation is there probably was a population crash. The modeling has implied, which is a disturbing one, is that if we don't do anything about HIV, maybe in a few decades or 100 years, humans will also evolve the coping strategy that pan-troglodytes has evolved, but we will have to undergo a population crash in all likelihood to allow that to happen. And we're already seeing a population crash in parts of Africa, where there's now sort of negative population growth based on what's happening.
What about other varieties of chimpanzees other than this particular one?
Right, so there are multiple subspecies. We haven't found this same virus in other subspecies. It seems to be, so far, it seems to be confined to pan-troglodytes, troglodytes.
How long do these pan-troglodytes live, and how do you know it doesn't slowly kill them?
Right.
Like people live 8 years asymptomatic and then die.
Right. So the question is, based on their lifespan, do we know that they actually live a full life, if you will, and are healthy with the virus? So pan-troglodytes, troglodytes live, I believe, for as long as 35 to 45 years. And based on the longest lived sort of specimens in captivity, which have been infected with SIV-CPZ, none of them have died from anything that looks like AIDS. They've died from other causes. For example, the first chimpanzee that was found to have the virus, she died in childbirth. There wasn't any complication from the virus itself.
What kind of percentages of your hepatitis are infected?
Well, so based on Hans' last published paper, which came out earlier this year, the evidence is on the order of 20 to 26% of the wild population appears to be infected with SIV-CPZ. But now what is troubling here is that nevertheless there appears to be a diversity of SIV-CPZ even in just pan-troglodytes, troglodytes, and zoonosis continues to occur. So for example, as new roads are built through the jungle in Africa, what we have is an example of what is called the bushmeat trade. So this is in fact a roadside stand on a highway in Gabon. And you probably can't recognize it, I'm sure, but what you're seeing here among this sort of remarkable array of exotic species which are available for purchase for food are two butchered bodies of pan-troglodytes, troglodytes chimps. So that what's called the bushmeat trade continues to occur, and this actually leads to increased exposure to SIV-CPZ continuing. And it also facilitates the movement of infection into urban areas. So that one of the challenges that we're facing is that we potentially may see new zoonotic transmissions into humans, and that unless something is done to eliminate the bushmeat trade or to at least limit it as much as possible, we have two risks here. One, pan-troglodytes, troglodytes will go extinct. If this chimp goes extinct, we will lose our opportunity to study what is in fact the natural host reservoir for HIV-1. And we may lose our only opportunity to understand how another primate lives with this virus. But the threat also remains that as additional zoonotic transmission occurs, we may in fact see the emergence of an even more pathogenic strain or type of HIV-1. And the epidemic is already the most daunting that we've seen in human history. I think it's safe to say that we don't want to see any further new HIV sort of epidemics emerging. So it's crucial that we try to contain this virus as much as possible. But what is quite clear is that it's very likely that the degree of threat that this virus represents is very much tied to the fact that it has evolved over probably thousands of years in a close battle with a primate immune system. And that's a very interesting take on the pathogen for us to consider. Question?
Is the transmission of the virus in chimpanzees the same as in humans?
Yes. Yeah, so the question is the transmission in chimpanzees the same as in humans? Yes. It is also transmitted by sexual contact and by body fluid exposure. Yes, and by blood exposure.
Do you know, or have they been able to find out, is it just like a given community of chimpanzees would be infected, but another given community would not be because of crossbreeding between those communities?
So so the question is it clear then that infection is contained within communities that potentially are united, if you will, by sexual relationships among the members? It's not clear yet. Once again, the problem is because they're endangered, there are just not that many of them. And the tracking of communities in the wild is very difficult. Because once again, there hasn't been sort of the systemic tagging of wild pan-troglodytes, troglodytes, and following them over time. That's being done now. So probably that will emerge over time as well. Whether or not it's very local specific in West Equatorial Africa, if there are actual communities of the chimp that perhaps have never seen the virus before, we don't know that yet. Because frankly, not enough is known about this particular subspecies in terms of, for example, how much cross-group sort of connection or interface or contact there exists in the wild. We simply don't know at this point, which is frustrating, because that would be a very interesting thing to know as well. Because being able to study the transmission dynamics in its natural host reservoir would be very important for us to understand as well. One more question?
Explain why it's such a low infection rate, 20%?
That could very well be the case, yes. But it may also be the case that it's a low infection rate because the number of individuals present in the wild are so low now that that tends to isolate sort of pockets of chimpanzees, one from the other. That's a very significant possibility. So, in terms of the virus itself, what's worth noting is that it is, even though it is clearly such a deadly pathogen, it is a relatively simple structure when you look at it. So that if you look at the structure of HIV, for our purposes, what we really need to worry about are what the so-called membrane envelope of the virus, which has studying it two viral proteins, GP41, which is a glycoprotein, and GP120. Those proteins are critically important for that initial molecular handshake that allows the virus to recognize target cells. What is also of crucial importance is contained within the core of the virus itself are two strands of viral RNA, that is the genome of the virus, and packaged with those strands is a viral enzyme called reverse transcriptase. That is a crucial player in, if you will, why HIV evolves at such a significant rate in the body of a single infected individual. So that, as you probably know, viruses in and of themselves have no capacity to replicate on their own. Of necessity, they must infect a host cell in order for them to replicate and produce progeny virions. So that one of the clear hallmarks of a virus, and it's the reason why viruses are frequently referred to as non-living or as pseudo-life forms, is that viruses have no ability to harness energy, to replicate, or to in fact build macromolecules which are essential for the assembly of new virions. So that viruses must harness host machinery in order for them to replicate. So that if we look at the example of an RNA virus, such as HIV, that genome must enter the cell in some manner. And in the case of HIV, that RNA genome must be copied into DNA. This DNA is then subject to transcription like any other piece of DNA in the nuclear genome. RNA is made, it's translated into viral proteins. That is what is essential for the assembly of progeny virus particles and the budding from the cell of new virions. So that quite simply, the virus must harness the central dogma of information flow in the cell in order for it to replicate. And HIV is no different from any other RNA virus and from any other retrovirus in that regard. So HIV is called a retrovirus really because of the critical role in the early stages of the life cycle of what is described as reverse transcription. So that once the virus enters the cell, the RNA genome must be reverse transcribed through a two-step process into complementary viral DNA. This viral DNA is imported into the nucleus, becomes integrated into chromosomal DNA, and is able to harness all the sort of regulatory mechanisms of the central dogma to produce protein. In this case, viral proteins from what is described as an integrated HIV provirus.
Does it integrate anywhere, or are there specific places in the genome where it integrates?
So the question is, does the virus integrate randomly into the genome? It integrates semi-randomly. In other words, there are hotspots in the genome that are particularly susceptible to the integration of the viral genome. The reason for those hotspots is that those are the sites in the human genome that are most exposed, which are those sites which are most transcriptionally active. So in some ways, what HIV is able to do is to piggyback on those parts of the human genome that are being heavily expressed, and as a result, viral proteins are heavily expressed as well. It's a very tidy sort of relationship. But HIV's ability to evolve sits squarely in one place and one place only, right here. That is where the process of evolution, or the foundation for evolution for the virus, sits. It is in the reverse transcription of viral RNA into complementary DNA. And that sits squarely, that responsibility sits squarely with an enzyme called reverse transcriptase, which comes in with the virus. It is packaged with the virus. And what's interesting about reverse transcriptase is that it is able to take the single-stranded RNA genome and reverse transcribe it into a complementary double-stranded DNA that's available for integration. It is a remarkable enzyme because it is made up of 2 subunits, but unlike most enzymes that we're accustomed to thinking of, it has 2 discrete active sites. One active site allows the polymerization of DNA. The other active site actually degrades the RNA genome after it's fulfilled its function as a template for reverse transcription. But what we're most concerned about is that as an enzyme, HIV reverse transcriptase lacks any proofreading activity. So as you know, DNA polymerase in your body proofreads in the process of DNA replication so that it corrects for misincorporated nucleotides. So that, if you will, the DNA sequence is maintained correct and proofread. HIV reverse transcriptase lacks any kind of proofreading activity. But to make things even more remarkable, on top of not having any proofreading activity, it is also remarkably prone to mistakes. So it has an error rate in terms of misincorporation of the wrong nucleotide of a rate as high as 1 in every 2,000 nucleotides. What that means is that if you look at the HIV genome, which is roughly 10,000 nucleotides in length, every time a copy of the HIV genome is made in DNA form, you can have anywhere from 2 to 5 mistakes made in terms of the wrong nucleotide being incorporated. This is one of the highest rates of error of any reverse transcriptase known. Because you might be asking, are all reverse transcriptases like that? If you look at other reverse transcriptases, they will have a rate of error of 1 in 30,000, 1 in 45,000, this 1 in 2,000, and in fact there have been some measurements that go as high as 1 in 1,500 nucleotides. This error rate means that the HIV genome is remarkably plastic. In other words, it changes all the time. And there's the remarkable diversification of the genome in the body of the infected individual. This is the basis for resistance to anti-HIV therapy. Because if you think about it, the population of virions in the body is remarkably diverse, remarkably quickly. So that unfortunately we're sort of running out of time. But a point that I want you to really understand is that HIV, the genome, is changing all the time. It's changing all the time. When you couple that with the fact that on the order of 100 billion new viruses are generated each and every day in the body of an infected individual, what this tells you is that you have this enormous population being produced every day that is remarkably diverse. You also have to couple this with what is a relatively short lifespan of the virus in the body. So short, in fact, that in the body of an infected individual, half of the virus is replaced every 6 hours. Every 6 hours. So what this means is that you get infected with one strain of HIV. By the time a few months have gone by, your body is full of scores of different strains of HIV. So that the body of an infected individual becomes, in fact, this remarkable ecosystem, if you will, of different HIV strains. So this, believe it or not, this sloppiness is the strength of HIV, because it allows it to be remarkably diverse in the body of a single individual. But given how high the error rate is, what percentage of virions do you think are infectious in the body? Let's say you isolated from the body of an AIDS patient 1,000 virions. Do you think 500 of them would be infectious? Do you think 900 of them would be infectious? I know some of you know the answer to this, but what do you think? What was that? Less than 1%. That is in fact true. In fact, less than 1/10 of 1% are infectious. So that don't think that this remarkable mutation rate is necessarily a good thing for the individual virus. The vast majority of virions are inactivated by this remarkably high mutation rate. But when you couple that with a replication rate that produces billions of new virions per day, there's still more than enough pathogenic infectious virus to cause disease and to have a broad enough population, a diverse enough population, to eventually see the emergence of a mutation that confers resistance to a particular drug. Question?
What I find fascinating is how it manages to hide in the host. I mean, I know that the host will set off a response to the virus, but at some point, and I don't know whether you had told us this before, the body no longer responds to the virus incorporating itself in the genome. And is there, what's the response to why the body doesn't see it continually?
So the question was, why is it that it seems as if for so many years the body does not see the virus? And in fact, originally it was thought that was the case, that somehow HIV hid out and was latent, which is why generally HIV is a slow illness that kills you after 7 to 10 years. In fact, your body never misses HIV. Your body sees HIV immediately. And in fact, your body wages a pitched battle against HIV each and every day from the first time that you're infected. So the human body never misses HIV. The human immune system never misses HIV. And in fact, the ability of the immune system to tolerate what we believe is on the order of 2 to 3 billion T cells being killed each and every day that you're infected with HIV completely opened our eyes to the regenerative capacity of the human immune system. So in fact, that clinically latent phase of AIDS, of HIV infection, is a pitched battle between your immune system and the virus. What happens is your immune system ultimately fails. It can no longer replace the billions of T helper cells which are killed each and every day. Gradually, the steady state number of those cells collapses, and the consequence is disease as your immune system fails. So somehow, troglodytes, we think, does not experience that failure. So somehow, the regenerative capacity of that chimpanzee immune system is very different from ours. And in fact, there have been some very interesting theories that for pan-troglodytes it might be a benefit to have this virus. That potentially the virus may may select for a much more high regenerative capacity immune system, which in the context of the equatorial rainforest, where you're exposed to many pathogens, may in fact be a benefit for pan-troglodytes troglodytes. That is one possibility as to why this may have happened over time. Our immune system is not like that, though. So our immune system does fail after 7 to 10 years.
Aren't there some humans that are asymptomatic after infection?
So there are populations, there's a very small group of what are called long-term non-progressors. These are individuals that do not appear to progress to disease. Some of them have mutations in a receptor, or actually a co-receptor, that is important for highly efficient HIV infection. Others seem to have other changes that we're not entirely aware of what they are yet. What is unfortunate is that several of those long-term non-progressors are now progressing to disease. So that disease appears to begin, instead of after 7 to 10 years, it appears to begin after 15 to 20 years. And so it's not clear whether or not there is a true human non-progressive state. It's not clear. The jury is out at this point. So in terms of resistance to anti-HIV therapy, boy, this is a little bit frustrating because I wanted to tell you about some of the mechanisms for anti-HIV drug therapy, because that would give you insight into how the resistance has altered, how the mutations have altered the virus, but unfortunately we don't have time for that. But what I will sort of leave you with is that one of the interesting issues here is that the first drug that was approved for use in AIDS patients was a drug called AZT. From the very beginning, it was clear that after roughly a year, resistance to AZT emerges in the body of the infected individual. The drug stops working. It was thought that the beauty of combination therapy, which, as I said, remains the gold standard for treatment, is that if you hit the virus with drugs that attack the activity of more than one viral gene, the probability of the virus evolving resistance in 2 different viral genes and still being pathogenic at the same time was vanishingly small. It was so unlikely that there's no way that could happen. And in fact, when combination therapy was originally tested, after the first year, no resistance. After the second year, no resistance. It's after the third year that resistance emerged. So lo and behold, my goodness, the virus could evolve resistance to both a drug that inhibits the reverse transcriptase enzyme and a completely different drug that inhibits the protease enzyme of the virus. So that 2 changes that both allow those enzymes to still be active, but yet be sufficiently different to no longer bind the drug, could appear. And what I think this perhaps underscores is just how diverse the population is. That with this high mutation rate, this huge population, that virtually anything sort of emerges in terms of what is possible in the genome of the virus, in terms of changes, subtle changes, so that even with combinations of 3 different drugs, resistance still emerges. So I think it really underscores how highly evolvable this virus is.
Does that have to do with repeat infections with different strains that the patients experience?
Right, so the question is, does that have any connection with multiple infections with different strains? No, not at all. What it underscores is, once you get one strain in you, you have legion. You have a legion of strains inside of you ultimately. And so that, I think, is really what is so remarkable. What is of concern, however, just so that you get a sense of what the therapeutic principle is now, the hope is that as we identify different types of HIV drugs, as resistance emerges due to evolution to one drug, you can salvage therapy by using another drug that has a completely different line of attack. And that if we have sufficient numbers of drugs with very different lines of attack, we can hopefully extend the period of relative health with HIV to well over a decade or more, 20 years, maybe 30 years eventually. The problem is, as new transmissions occur, HIV-infected individuals are transmitting strains of HIV that are already drug resistant, which means that if you acquire a strain that is already resistant to X, Y, and Z, you will lose the benefit of the 2 years, 3 years, 4 years of drug therapy that you might have had otherwise. So that is another reason why it is so crucial that we try to limit further transmissions of HIV from the population of infected individuals that are currently taking drug therapy. So that's why it's crucial that even when patients are healthy and their combination therapy is working, that has very little or reproducible correlation with the amount of virus, for example, in genital fluids. So that you could potentially have a low amount of virus in the bloodstream due to combination therapy and be perfectly infectious in the context of sexual transmission, which is why for patients on combination therapy, it's crucial, for example, that safe sex practice be adhered to. So, sadly, I have run out of time. I wish I could join you for dinner, but as I hope you all know, I have a class at 5:30 on Wednesdays, which is why I haven't been able to join you this entire fall. But once again, we have been honored, absolutely honored having you. And now you're going to hear a much more interesting talk from Andrew Murray. But I will pass things over to Tara to introduce Andrew. But thank you.